Hi, guys. All right, let's see here. Let me answer this text. How are you guys? Oh, no.
I'm sick.
Good.
I have COVID since Wednesday. What was that, the day before Thanksgiving?
Cold or COVID? Which one?
COVID.
Oh, no. Oh, I hope you feel better. You're the 10th
Yeah.
person today that I've spoken to that said that to me.
Oh my gosh, yeah, it sucks. It's really like a cold, but the part that sucks the most is this freaking dry cough and not being able to smell.
No, absolutely. My daughter got a false positive right before Thanksgiving, but I knew that it was a false positive. So what I ended up doing is at the time I wasn't certain. So she was like in exile. Like I set a little table for her. I moved everything outside and she was all the way on the other side of the yard. She felt like typhoid Mary, but that's okay.
Oh wow.
You guys, last test and we're all done. Literally
Yay.
last one. No, not yay. You mean you're going to miss your favorite class? No, yay.
Yeah.
We're gonna miss you for sure.
I miss you, not the class, but you.
Thank you. You will miss the class. All right. So let me screen share. I want to tell all of you about what is going to be on the exam question wise so that you know sort of what to focus on and where to put your energies. So actually this one, you guys, someone show me a thumbs up or something. You can see the screen share, right?
Yeah.
Okay, thank you. So you're going to have 4 questions on knowing the components of a virus. So if you were to look at this picture, the first one you should know is enveloped, and the one on the right is naked. The line around the one on the left is the envelope. The pink and yellow little lines and arrows on the bottom, those are the glycoprotein spikes, which you're hearing a lot about right now. So when you hear that COVID has mutated and Omicron and all this other stuff, it's this pink and yellow, it's these spikes that are actually changing. There's the genetic material inside, which it's important that you realize it can be RNA that's double or single stranded. It can be DNA that's double or single stranded, which is strange because traditionally in bio classes, DNA has always been double stranded and RNA has been single stranded. I bring this up because if you were to hypothetically be asked a question of how do you know if it's by looking at a picture, if it's DNA or RNA or single or double stranded, and quite honestly, the answer is you don't. There's no way for you to know if it's double or single stranded unless if you are specifically told, or it happens to be a specific virus that you are expected to know, like for example, influenza is an RNA virus. Otherwise, you cannot tell by looking at it. Each of these individual little balls are known as capsomeres, and they make up the capsid, which is a protein outer lining. It's internal to the membrane, but this capsid that's made of protein is actually protecting what we'll just call the genetic material inside of the virus. So please know that there are enzymes within the virus. They're called proteolytic enzymes. That's not as important, but these enzymes are within the virus that help break down the capsid, because in order for this guy to replicate, it's going to have to get out of not only an envelope, if it has one, but also the capsid, which is that protective protein coat. So 4 questions about the parts of a virus. And what we've just been talking about covers about 5 things already. And then this picture, one second, let me turn this in the screen. That looks really annoying. There we go. That looks better. Or maybe it doesn't. You can tell me if this looks better. I'm trying to get it full screen. Okay. Is it full screen? Should be. Okay. So again, you have enveloped versus naked. They both have a capsid. Capsids are made up of individual capsomeres, and inside is the genetic material, which is either DNA or RNA. And unless if you're told, you absolutely don't know. In the top right, I don't expect you to remember any of these, but I do want you to be aware that viruses have some pretty strange shapes. The bacteriophage is like a little lunar landing. Some of them are round and coiled. For example, rabies looks like a stack of pancakes. Traditionally, bacteria are just staphylococci or Vibrio, spiral, or rods. They don't have all these odd shapes like viruses do. And know that viruses are incredibly small. They're measured in nanometers, which is a smaller unit than micrometers, which is how we measure bacteria. This picture is one single E. coli, this green thing in the middle. And inside of that, you see 2, 4, 6, 8, 9 viruses fitting inside of one rod of E. coli, just to put it in a visual perspective for you all. So besides 4 questions on parts of the virus, there are 8 questions on the viral life cycle. There's one picture of the viral life cycle that looks like the one in your PowerPoint slide. And then after that, you will see that there are, oh, hold on. Let me answer this thing in chat. You guys can hear me, right?
I can hear you.
Okay.
Yeah, we can hear you.
Let me answer this student. I'm just going to tell them to log out and log back in because I know that it's working. Okay, so for the steps of the virus life cycle, you're going to see one picture. It'll be labeled A, B, C, D, E. What's happening here? Is this enveloped? Is this naked? That kind of thing. So 8 of those. To go through the steps, first is attachment or absorption. So here you have the viral particle. Is this enveloped or naked?
Envelope.
Exactly. Perfect. If this gray portion was missing, the capsid would be the outermost part, and that would be a naked one. Notice these green little things sticking out are the glycoprotein spikes. And on the other side, this darker gray looking thing is actually the whatever it's going to attack. So say lung tissue. There are receptors on the host organ, membrane, or cells that this virus is going to attach to. So the first part is either attachment or absorption. They will both work. Next, this is that single or double stranded. Remember, it's unique. So let me open every picture here and then go through it with you. Expect to see a more cartoon animated version of this picture for the virus life cycle. So first step is again attachment or absorption, where the virus attaches to the membrane of whatever the host cell is. So if it's a respiratory, assume lung tissue. Then once it gets inside, the term for that is penetration. This is the virus penetrating into the host cell. And then we have uncoating. This is where that proteolytic enzyme is going to break down not only the membrane, if any is left, but also the capsid, which is the protective protein outer coating. Once that occurs, all of the components happen within the host cell because a virus is an obligate intracellular parasite. It's not alive of its own right. So it's using the host functions to do replication, transcription, and translation. It does these very quickly, and it tends to do whatever the host is doing since it doesn't have its own machinery to go through all this gene expression. Once that happens, the next step is assembly, which is also known as maturation. Knowing me in your lecture videos, I will have said something like it's building its little army because it's now assembling all these viral particles because the last step is release. Release is when it's now been replicated, transcription and translation have occurred, and it's impacting the neighboring tissue and cells. Your picture is going to look 80% like this guy on the left here. Maybe I'll just show you it quickly later. But again, 9 questions on just the life cycle. So this guy right here. Another thing I'll tell you is virus information. There's 10 of those. So true or false, all viruses have RNA as genetic material. Perfect. 20% of viruses are not dangerous and pathogenic. What's the right answer, Daniel? No. Don't correct yourself. You were right. So I said 20% of viruses are non-pathogenic. Is that true? So what's the answer? What percentage of viruses are non-pathogenic?
None. They're all pathogenic.
Who said that? Terry, was that you? I don't know. Somebody
That was
said
them.
that's the right answer. They are all pathogenic. There is no such thing as a good virus. It's the opposite of bacteria, where close to 90% are good for you. Short for cytopathic effects is CPE. So again, viruses are not alive of their own right. They need a host. So when you work with things like tissue culture, you need to grow it on some type of live cell. Traditionally in a research lab, they'll use MDCK or HeLa cells. There is a movie about Henrietta Lacks, and that's short for HeLa. It's her cells that we're using a gazillion years later. The family keeps suing for money and they never get it, but they're owed that money. The other one are MDCK, which is Madden-Darby kidney cells from a monkey. So the point is, it's always got to be grown on some type of live tissue. You need to know there are 2 cytopathic effects that we discussed that could occur with viruses. One of them is inclusions. Inclusions are holes. So if you look at this picture, all these little white things are holes within the tissue that are made by the virus that's infecting the host. And the second one is syncytia. This is the synergy of 1 + 1 = 3. This is the viral version of a biofilm where they all stick together in large, big groups, and that's what causes them to be even more pathogenic. In young kids, RSV or respiratory syncytial virus is a big deal. Not so much in us, but in younger children, especially toddlers and under the age of 2, we'll say. So here what happens is the virus gets in, makes these large plaques, almost like biofilms, but they're called syncytia within the lung tissue to cause the illness to be more dangerous for them. And then we'll go past some of this for a second. Okay. So there are 9 questions on prions and viroids. 9, and there's only 2 slides on it. So I'm probably telling you that's 20 points on your exam. Please just make sure you know these. You're going to see the question saying, know the information about prions versus viroids. And the answers to each one is it just prions? Is it just viroids? Or is it both? So what's both is both of these guys are just one single component. It's not like a bacteria where it has all the different components inside. It's just one thing. So prions are nothing more than proteins. And what is their pathogenesis? They're folded incorrectly. And this abnormal folding, it occurs at chromosome 20. You don't necessarily need to know that, but I want you to know it causes diseases that are fatal. So spongiform encephalopathy, that is putting holes like Swiss cheese into the brain is what it does. Mad cow disease or Creutzfeldt-Jacobs, it's called CJD for short. Those are the 2 main illnesses. That is all you need to know on prions, and it is a large chunk of your exam. This is it. So make sure you know prions well. That's just a picture version of it. And same with viroids, also made of only one single thing, RNA. There's no nucleus, no mitochondria, no protein. There's nothing but RNA. And what it doesn't even code for anything. This guy causes plant diseases, so not in humans. And its pathogenesis, meaning how does it cause it or what's its infectivity, it interferes with translation. And you know that because with RNA, you always make protein, and in viroids, you don't. A big mistake that students make on this exam, so I want to make sure that I point this out, is a viroid is this thing in front of you right now on the screen. A virion is, so I'll spell it because I'm watching the transcription on the bottom. It doesn't look right. It's VIROIN, or or ION, I should say. A virion is a complete viral particle. So when you look at the life cycle and it says the assembly and maturation, that's a virion, not a viroid. So just make sure you're not using those two terms because that is relevant in the exam. So virus host interactions, there's 3 terms here that I want you to be comfortable with. First one is acute. Acute means that the climax of the illness develops super quickly. The symptoms are rapid onslaught. So if you catch the flu, basic influenza very quickly, that is an example of acute. Hepatitis A is also an acute type of virus. Chronic, on the other hand, hepatitis B is a good example of that. This is where it's ongoing. If we were talking bacteria, Mycobacterium, or the bacteria that causes TB, that's considered chronic. So over a long time, this thing lingers. It's multi-drug treatment to treat the person. A long convalescence means that that individual is recovering for a long time, but while they're recovering, the virus is still shedding, so they can still be infectious. And then the last one is latent. Another name for that is dormant. It basically means the virus is sleeping. HSV is short for herpes simplex virus. This is where viral shedding is occurring and you can still be infectious even if you were to test negative for it. HSV is one of those viruses that sort of like the gift that keeps giving. It doesn't just go away. Once you have it, you actually have it. For the virus portion of this exam, know that it's a lot simpler than bacteria because you don't have to worry about the spelling. You don't have to write influenza. You can just write the flu. You don't have to write respiratory syncytial, write RSV or HSV. You can use all of the shortcuts. You don't have to write out the names of anything, and there's no underlining or any of that. This is just a picture view of acute, chronic, and latent. So make sure you take a look at that. Don't worry about this slide at all. Oh, that was quick. Hold on a second. Let me pull up the other one. Where is it? Here. So there are 3 questions. Oh, great. Wrong one. There's 3 questions on cancer. The questions on cancer are relatively basic because it's not something that I asked you to do a whole lot of work with. I'll give you an example. See if you guys can answer this. The study of cancer is called, I'm asking, someone tell me.
Oncology.
Perfect. It's oncology. There's 2 types of tumors. There are benign and malignant. If you have to have one, which do you want?
The benign.
Exactly, because malignant are the ones that are cancerous. And malignant tumors tend to metastasize. That word means that it moves. So a woman who is diagnosed with breast cancer, they will instantly check right by the lymph nodes near their armpits because it tends to travel. And that's a very common place that breast cancer tends to travel. So metastasize, benign, malignant, oncology, basic things. And know the definition of a proto-oncogene. So oncogenic genes are cancerous ones. Proto-oncogene, we all have those, and they either do or don't turn to cancer. The way cancer works, it's similar to viruses, which is why it's in this particular unit. And what's strange about it is that there's not a lot of understanding. Many of the cancers are not easy to treat. Right now, testicular, prostate, and breast cancer are a lot simpler to treat, but usually in the GI tract. And when you are talking about types of leukemias, because that is cancer of the entire blood, those are very hard to treat, hard to catch by the time a person becomes symptomatic. Unfortunately, at that point, their bodies have already been overrun with cancer, and most likely it's difficult to survive. So those few little things about cancer and nothing more. I want to go over this second PowerPoint. There are 9 questions related to this second PowerPoint. Similar to the bacterial pathogens, I can't really say the viral pathogens because all viruses are pathogenic, right, Daniel? Every single one bad. So I ask you questions like, what organ system does it impact? Or something possibly unique. So let's talk about a few of these, but I really, really do want you to rewatch the lecture if you have time for the second part of Unit 5 PowerPoint, because every single thing in those 9 questions is covered 10 times more than you even need for the exam. So if we start, what's going on here with influenza? Influenza happens to be an RNA virus. A lot of similarities to COVID. That's why in the beginning, a ton of people kept saying, oh, it's just the flu. No, it's not. But how do you get it? Direct contact. It's airborne. It's respiratory. If I choose to lick Lisa and spit on Mo and start licking Terry and breathing real close on Daniel, you're all at risk. If I have influenza, it is transmitted through air. It is direct contact, highly, highly contagious, and a very high mutation rate. Most of you already know this because when you get a flu shot and when they start talking about them, which began in early October, that's the start of respiratory season, you will hear probably by mid to late December in the news. If you watch the local news, they'll tell you the efficacy or the percent that this year's flu vaccine is helpful. Why do they do that? Because it's a statistician program. They don't know. They look at numbers of over the past 2 decades. This is the type of flu that was most prevalent, and they concoct a flu shot or a vaccine, an immunization. Why? To help you. Does it keep you from getting it? No. But if you were to become ill, the symptoms are lessened, kind of like what you're seeing with the COVID vaccine right now. This picture is just to show you that they have an insane high mutation rate. That is the special information about influenza. Another thing to consider is it jumps from animals to people. SARS, if you remember, severe acute respiratory syndrome, that kind of started along these lines. So all you need to see here is that it jumps from humans or animals to humans. Most have been isolated in humans. And if you look at the top left, from quail to geese to fowl, doesn't matter which one, all of them essentially. When you hear H1N1, I mean, that has to sound at least familiar to some of you. Every year you're going to hear the H's and the N's. If you refer back to this picture in the PowerPoint, if they tell you this year it's H9N2, it's very prevalent in quails. So it's kind of interesting. What are the H and the N? It's hemagglutinin and neuraminidase. These are those spikes that keep mutating that you hear about with this new variant of COVID as well. So COVID and influenza have a lot of similarities in how they work as viruses. Every year, the strain changes. How birds migrate determines the severity and the virulence of influenza that particular year. So as a student told me last semester, we should just kill all the birds and the ducks and the chickens and we won't have influenza. But since I like to eat all of those things, I'd prefer not to kill them. So put that out there. I am hoping nobody needs me to define the word pandemic, and I'm not going to. You've all heard about it. You've heard talk of the Spanish flu. In a few years, you will see, if you ever hear about pandemics, COVID's going to be real high on that list of conversation. When something is endemic, that means it is more localized to a specific region. Pandemic is, it's now worldwide and global, which is exactly what we're seeing. RSV, this is huge in young children. Usually between 8 and 15 days, it's very prevalent and then it can go away. The young infants and the younger the child, they are symptomatic of pneumonia and they have a high swelling and get an obstruction in the areolar and the bronchioles of their lungs. So this is respiratory. What tissue is infected? Lung tissue, everything related to the respiratory system. Again, this is respiratory and therefore highly communicable, meaning passes from person to person very easily. And it is also a high mutation one. HSV. One second. I'm in the middle of a student review. I'll call you soon.
No problem.
Bye.
Bye.
So with herpes simplex virus, you've got type 1, type 2. These are 2 very different kinds. A couple terms that attach to them is sequelae and latent. Latent is again like dormant, that this guy's sleeping. And sequelae basically means that in this situation, they something else can trigger it to come back and wake up from its sleep is the simplest way to explain it. There are many different kinds. Lesions are one of the symptoms that a patient with HSV will acknowledge in various parts of the body, everywhere from finger to mouth. That is the buttocks. That's the shaft of a penis. Here you can see the little red dots. So incubation's quick, 2 days to a week. So in a very short amount of time, you are in fact able to see the symptoms of this virus. HPV, which is human papillomavirus. There are many, many different strains. 2 of them, Gardasil is a vaccine that is out, which protects against strains 16 and 18. And again, there are hundreds of different kinds. These 2 are known to lead towards cancer, which is why it's relevant. That is an STI, and it is associated specifically with cervical cancer. So with HPV, again, incubation could be as low as a week, but check this out. It could be up to a year. So HPV is an illness that you may or may not know right away that you have contracted. It's incredibly common in people under their mid-20s. And urogenital tissue, anything that is an STI, the tissue it impacts is urogenital. You want to say reproductive organs, however you want to word it, it's that region of the body. This picture fascinates me because the head of this penis truly looks like a cauliflower, and I eat a lot of cauliflower, so it kind of bothers me. Please understand, these are, it's true. Look, it's kind of odd. So this is super extreme. Like, please realize you don't get HPV and you see this. That's not how it happens. The picture on the right is actually a vaginal opening. So, I mean, male and female alike, urogenital tissue, 3 out of 4 people in that most high level of sexual activity age, which is 15 to 49, have been infected at some point in their lifetimes. There's different levels of HPV. This is common, strange, and rare, but pictures got great shock value. So I have it in my PowerPoint. Rotavirus, this one, anyone with children will know what it is. Here, let me open up. Why isn't it opening? One more. Trying to get it to open more. Okay, so this one is the easiest way a child under the age of 2 can die from what is considered life-threatening diarrhea. It isn't diarrhea or that expelling of so much excrement that will actually cause the death. It's more so dehydration and loss of electrolytes and minerals. So super, super acute, which means the symptoms come very quickly. Everything you would associate with something like food poisoning, so vomiting and diarrhea, that is what causes the crazy dehydration that could become death. It could go to death. There's about 20 to 100 deaths a year in children under the age of 2, but over 3 million cases. So when kids play at the doctor's office and the mall and all those little places, that is where they're picking up rotavirus. Daycares, it's very common because it is extremely difficult to clean this virus up. A basic Lysol wipe doesn't really do the trick. So there has to be a very high level, intensive and heat irradiating kind of cleaning to figure this out. There's not a child on the planet that will not catch some version of this. So it's not super uncommon or anything like that. Okay. Oh, that last thing. I don't know if that's important. I don't remember, but I don't think so. Let me open it. So subsequent infections decrease. In other words, when the child gets it again and again and again, each time they build up an immunity to rotavirus, and it's less severe, less diarrhea, less reaction moving forward. So hepatitis, this one's interesting. Know that Hep A can cause acute foodborne and waterborne illnesses. So Hep A is related to food. It affects the liver very much. It's found in all blood and bodily fluids of an infected person. And then we have rabies. Rabies is endemic, so or more prevalent endemically. There are certain animals, it's very rare that it's in a domestic animal. So even if your dog's being psycho and you say your dog is rabid, chances are it will not actually have rabies. What I want you to know about this is it takes an animal that is infected to bite the person, almost like a vector, remember that word, to infect the person. If you look all around this picture from 1 through 6, what you should acknowledge is there's almost not an organ system in your entire body that is not impacted by rabies. It's painful. It hurts at the muscle sites. You have muscles from head to toe. Notice it talks about the eyes and the kidneys and respiratory and nervous system because the brain becomes infected. So when you're asked to name a virus that impacts a lot of organ systems in the bodies, this is a perfect example to consider. And this is how it's distributed. West Nile, what's the vector for West Nile?
Mosquito bite.
Yep. Come on, there's that cool flying thing up there in the picture. Absolutely, mosquito. It's becoming more and more prevalent here right now. There have been, I think, 7 people this year that have died of West Nile, which is super rare. Usually it's maybe 1 a year. So the mosquito issues have been a lot worse. And they're saying it's because we had a larger amount of rain this particular season. I'm not going to say much about that. Epstein-Barr. This is known as the kissing disease. Another nickname for it is mono. This is the first virus that I gave you a nickname for, and it's the only one. So maybe you want to remember that. Epstein-Barr, super common. The signs of mono or kissing disease or EBV, fever, fatigue, sore throat, swollen glands. Right away, when you read that, you should think, wow, so is the common cold, so is influenza, so is 1,000 other things. So often this becomes a little bit missed. The telltale sign that an individual has Epstein-Barr or mono is their level of tiredness. And I think I talked about this in your lecture. If not, I'll just mention it quickly now. Imagine you're sitting on the couch watching something good and you have to use the restroom. You might sit there and cross your legs for a little bit because it's a great part of your TV show or movie. But with mono, you are physically not able at times to get up and take yourself to the restroom. Just the thought of physically moving yourself is an impossibility. So that kind of tired, that overwhelming, just can't deal with yourself kind of tired. And when that level of fatigue hits in, this is the first thing they're going to test for. Like first thing always. And CMV, make note that CMV is pretty ugly. It's an incredibly common congenital infection, the most common, meaning passed from mother to baby. It impacts every single organ system in the body, and usually it's pretty asymptomatic. So this is most dangerous for a pregnant woman to contract because it often causes fetal demise, which is death of the baby that she's carrying. So, and again, it stays dormant in the body. It belongs in the herpes virus family. You don't necessarily know you ever have it because it is asymptomatic. But again, every single organ system in the body can be impacted with this particular illness. So there you go. Ability to infect lots of tissue types. It can cause all kinds of infections, and there are 3 examples of them. So I want to tell you one more thing since a couple of you came in late to make sure you have this information. So there's 4 questions on components of the virus. So you have a picture of a virus and then there's the components. You have 8 questions on the life cycle. You have 10 basic virus questions, which are things like, what are cytopathic effects? The answer is inclusions or syncytia. Or what unit of measurement is used to determine the size of a virus? The answer is nanometers. Or a true or false, all viruses are RNA core. No, they're not. There's DNA and everything else in between. Then again, there's 9 questions on prions and viroids. Do not mix up viroid with virions. 3 questions on cancer, super basic, as I had mentioned, because we didn't focus on that a lot. So the study of cancer, the 2 types of tumors, which are malignant or benign. And when something metastasizes, that means there's movement within the tumor itself. And finally, there are 9 questions of the second set of PowerPoints, which are the actual viruses themselves. Remember, you can use abbreviations. So that part is a lot simpler than it would be if they were bacteria where you have to name them all. This exam tends to yield super high grades. I don't believe this is a difficult one. It's a couple questions about things like the syncytia, and the inclusions. I do want to take a second to show you a picture of the life cycle in the virus, just as like a thank you for showing up kind of thing. So let me close this for a second. I don't know how, but I will try. Is it still screen sharing? It shouldn't be, is it? No, okay. Let me just see if I can open. Wow, I almost kicked you all out of here. I'm sorry. That could have been an interesting problem. I did that earlier today. I was running a really important meeting. I was the host and I kicked everybody out of it by mistake,
Oh,
not intentionally. And then I was like, wow, I don't know what happened to my computer. They're like, Jackie, you did that. I'm like, yeah, kinda. It was unintentional. So I kind of want to give you guys a little cheat here because I'm appreciative that you showed up. See this picture? This is question number 1. You're going to see this picture for 4 questions. What is number 1? What is 2? What is this called? And then let me show you the virus life cycle. There it is.
Oh.
So can you see the pointer when I'm screen sharing? Yes?
Yeah.
Okay. So here's the virus. Is this enveloped? Absolutely. Is it DNA or RNA? Henry, what do you think? I didn't know you were there because your faces were hidden. Is it DNA or RNA?
Let's see. I'm gonna guess that it's RNA. I don't know. It doesn't really say on there. And I remember from the PowerPoint you saying that like we can't just say unless it like specifically says.
And that's the right answer. So don't
Okay.
guess. The answer is if I don't tell you and it's not written and it's not, you tell me what influenza is because that's the only one I tell you guys. It's RNA. The answer is I have no clue. We just call it genetic material. Perfect. So here's our genetic material covered by capsomeres making up the capsid, which is a protein coat. And here's your glycoprotein spikes. Step 1 is attachment or absorption. And then penetration is happening right here. A is basically you're uncoating, you're opening it up. Then all your stuff is happening in the middle there, replication, transcription, translation. And then when you get to the bottom here, what's happening is your little army of assembly and maturation. And finally at H, you see release, where it's releasing the particles. So I guess I'll show you what, is this naked or enveloped? Which is it?
envelope.
There you go. So you will see this picture 8 times. Okay. So I'm telling you, know the virus life cycle. Prions and viroids, just basic questions. You have A, B, or C. It's prion only, viroid only, both. Those are your choices. So the only thing that's really both is they're both made of just one thing. One is just protein, one is just RNA. Yes, there's going to be several short answer, but hold on. I'm making sure I'm not screen sharing now because I don't intend to.
Yeah.
Hold on. One related to size, because remember, what's the unit for viruses?
Nanometer.
Okay.
Nanometers.
You know that conversation that Henry and I just had about is it DNA or RNA?
Yeah.
That's another one. That's a essay. Well, I'm not going to say essay. You know, I hate those. It's a short answer question.
Short question.
There you go. One of those. I could ask you pathogenesis for prions and viroids. That's always an option. But other than that, there are no more. No, that's it. So
It's
one
only like
related to
four
size and one related to what Henry and I were chatting about.
Any true or false?
Yeah, there's a couple. That goes to the part where I said like, oh, wow, it's getting dark. Sorry, I'm outside. I don't have a light. Let's go into where there's questions about virus. Like when I was bugging Daniel before saying, hey, how many viruses are 20% good for you? So if I say 20% of viruses are happy and beneficial to humans, would that be true or false?
False.
Those kinds of things. Those kinds of things.
Okay.
So you said there's only like 2 short answers, right?
There's 4, but really 2.
4. Okay.
4, but really 2.
All right.
2 are the ones I told you, and I already told you you have to know pathogenesis for prions and viroids. But please
Okay.
understand, I'm looking for like 3 words. Do not write me a book. I don't want a book. I don't want to read a book. And the more you write, there could be something wrong in it. So just simple, simple. This exam is very successful for any class in micro I've ever taught, university, community college, any school, anywhere, any year. So please study hard for this one. It is our last thing in the entire class. You'd be insane to not do the extra credit. It's 5 points, half a letter grade. Why not? Several of you have already turned them in. I graded your thioglycolate lab. So other than extra credit in this, that's all we have left. So put your heart into it. It's the last thing. Okay.
Okay.
Any other questions? Wow, you can't even see me. That's not half bad though. Sorry. I'm outside. There's no light out here. I get up and turn the light on, but you'd see I have on pajama pants. So with little cows on them. So I'm just going to stay sitting here. You guys have, actually, no, now that I told you, I'll show you. Look, they're really cute. They have cows all over them. I literally have on pajama pants all day in every meeting, except I put on a suit top for the other one. So please, are there any questions? Ask away. If you forget something and you remember later, just email me. I'll reply to you quickly. Be comfortable, study the PowerPoints. This exam is directly from the PowerPoints, even more so and easier than the other exams were. And there's a couple special Jackie questions in there that I know you will all enjoy. Okay. Well, have a wonderful night. And
Thank
please,
you.
and you're so welcome. Any questions, reach out to me.
Okay.
All right.
Okay.
Thank you.
You're welcome. Bye,
Bye.
guys. Have a wonderful
everybody.
night. Bye, everybody.